for actually for being here. It has been a long day, and we really appreciate that at the end there are some people still sitting in the audience and very much then interested to these talks. I just want to start by saying that being here from this morning, I have heard at least 3 or 4 times the word bioprotection. And so my talk is going to be a little bit introducing what bioprotection is and also going into some of the results that we got into this application with Christian Hansen, because we have been using a strain of Lactiplantibacillus plantarum for bioprotection reasons. So when we talk about bioprotection, the amazing thing in science is that nothing is new and nothing is like created. You know, it's like the matter, because we somehow in cycles we reconsider aspects that have been introduced a long time ago, and we give them a new perspective. And this is actually what is happening with bioprotection, because bioprotection was first introduced in 1996 by Michael Stiles from Canada, that coined this new concept of using the activities that microorganisms are exerting by metabolic point of view, but also by simple presence, against some of the spoilage and some of the safety issues that we have generally in food. And at the beginning, bioprotection and biopreservation actually was really focused on the use of microorganisms and their metabolites against foodborne pathogens. And so we have several examples of bioprotective strains that are being used in the past, but also in present against some pathogens like Listeria monocytogenes, for instance, in dairy products, but also in meat products. And in the last years, this concept have been extended also to the spoilage processes. And so we are now working pretty much towards looking for microorganisms that are able to produce antimicrobial compounds, able to, I would say, diminish the speed or decrease the spoilage process. And when we are talking about spoilage processes, as you can understand, the concept is much more complicated, because if we have targets towards food safety, then we know very well which the targets are. So we know that there are some foodborne pathogens that have to be inhibited. And so it is simple somehow to look for something that is able to control those foodborne pathogens. But when we are talking about spoilage process is actually a quite complicated process where there are a number of microorganisms that are responsible for it. We have bacteria, we have yeasts, we have molds. And so trying to move from safety to spoilage, there is a rather high level of complexity that we have to manage and we have to govern. And somehow this is what is happening in wine, because when we're talking about wine, we know very well that the number of the microorganisms for sure are or is much more limited in terms of diversity with respect to other foods. But no matter what, we have several ones that we have to take into consideration. And so by going further into the processes and into the competition mechanisms that actually can be exploited when we are talking about bioprotection and biopreservation, here are some of the examples of the capabilities of the microorganisms that we can use also in wine in order to control the populations that we don't want to have. We don't desire to have them in the wine. And so here we can talk about organic acids, because they are absolutely very good and exceptional antimicrobials. We have hydrogen peroxide, nutrients competition, but already the space. And actually this is a very important concept that we can extend in wine, because in wine and in must, because if in wine and must we have a major population that is able to compete and is able to control somehow what is happening in that matrix and what is happening in that environment, we are already on an okay situation. If we don't have a strong competitor over there, then the problems starts to come out. But then we have, diacetyl, killer compounds and killer factors, very popular in wine. But then we can also talk about bacteriocins, and talking about lactic acid bacteria. Lactic acid bacteria, for sure, are one of the groups that have been investigated more deeply in terms of producing these toxic compounds. So when we are looking at wine, actually by chance, I was invited to give this presentation just when, right when this paper that we have written with my collaborators in Turin, talking about bioprotection in winemaking, came out. So this is a picture that I want to show you because bioprotection can be really studied at different levels, because what I'm going to be showing to you today is actually the level of the in vitro competition, because as I'm going to be presenting to you some of the results that we obtain is all at laboratory scale, but it gives us already some very good indications that we can move and we can upscale the volumes of the mass that we want to ferment. But then we have already some good examples of bioprotection or competition mechanisms that can take place on the grapes. And here you have different examples of yeasts. And I can point the screen and, let me see, which way? With this one? Okay. Yeah, it's a little bit delayed, actually. I prefer to, but anyway, doesn't matter. So you have examples in the grapes. And here you have some, for instance, some yeast that are able to produce some antimicrobial compounds towards mainly filamentous fungi. But then we have already some experiences in real wine fermentation, so in tanks, And so, but just for give you like a very simple concept, when you are talking about bioprotection, already Saccharomyces can be a very good bioprotective agent. He's the best microorganism that has been adapted because he's able to produce alcohol, he's able to consume very fast nutrients. So this is exactly one of the best microorganisms that we can consider as a bioprotective agent, okay? So when we go into bacteria, and this is a little bit the focus of this presentation and also the following one, if we take into consideration the two main species that you have already heard today in the presentation by Marette Dutoit, Lactococcus, Lactobacillus plantarum, and Oenococcus oeni, you can see here that they have a lot of features that are making them perfect examples of technologically important microorganisms, because they can be really used as a starter culture, but then they can also be considered as a bioprotective agents because they have both the somehow passive strength but also the active strengths. So they can both actively participate or act as a bioprotective agents through the production of toxic compounds, lactic acid, but also bacteriocin. Now it is stuck, this dot there. Okay, it moved. So, but then also, they have the very positive strategies, like, for instance, the capability to consume nutrients or the capability to form biofilms. Biofilm formation is not an activity that we are pretty much interested in wine, but, they are able to actually organize themselves in biofilms. So in this context, it freaked out, eh? Sì, il bottone sotto, il bottone sopra. Ok. Okay. So we are living in a world that we know very well that has some challenges that need to be addressed, need to be faced. We have already heard today some of the strategies that we can use in order to make the wine processing and wine production more sustainable. For sure, there is one aspect that makes wine production probably into the spotlight or under the spotlight of sustainability, that is the use of sulfur dioxide. So sulfur dioxide has entered, like one of these compounds that have to be absolutely canceled from wine production. However, we know very well that probably the bigger quantities of SO2 are not actually used in wine, but they are used in other food products. But anyway, now the focus is on wine. And so when we're talking about sustainability, of course, we need to make our processes more sustainable. And the decrease of SO2 is for sure one direction to go. But when we are decreasing the SO2, we have a lot of other problems that are actually coming out. As a microbiologist, I can tell you that some of the defects or some of the wine diseases, because in Italian we call it as malattie del vino, wine diseases, just to make very important the fact that when wines are spoiled by bacteria, they are like, they have a disease. So those diseases that were completely eliminated by the introduction of SO2, nowadays they are coming out again. They are coming out again, why? Because we are decreasing the SO2, we are not really changing the way we make wines, and so we have contaminations and we can have problems of wine diseases again. So in this context, there is, this possibility of actually exploiting the antagonism of some of the microorganisms that we can have commonly found in the winemaking. And one of those is, again, Lactobacillus plantarum. And so a couple of years ago, we were contacted and we had this actually a lucky circumstance to get in contact with CHR HANSEN, that they had this nice product that they wanted to test, and they wanted to understand a little bit better, what is at the basis of the mechanism. So they had this product that actually was suggested as a malolactic bacteria, but when bioprotection started to become hot, then they also wanted to exploit it further. So what we have done was to actually use this strain in laboratory conditions, just mimicking spontaneous fermentation, where we also helped a little bit the process with addition of SO2. So here is the experimental setup. I hope that you will be able to see it. So what we have done was to reconstruct a spontaneous microbiota formed by non-Saccharomyces, mainly Hansenia, but also Microbiology and Starmerella. We added a little quantity of Saccharomyces cerevisiae because, again, we wanted to get close to what is happening in the real world. And then, in some conditions, we also used small quantities of SO2 with or without the addition of our bioprotective agent. Here you have the experimental setup. So we have run it in two subsequent vintages, in 2019 and 2020. We have tried to maintain the conditions of the mast as similar as possible. So in the second vintage, we have corrected a little bit, both in terms of a quantity of sugars, but also in the final pH, in a way that we could have similar results. And then we ran those experiments in laboratory conditions, looking at the microbial counts and also the chemical composition of the wines. And we did not only basic analysis, but we also went into volatiles. And so we ran some GCMS in order to understand also the impact that those practices have on the final quality of the wine. So here are some of the results that we obtained for 2019. You see the different theses, from the control to the one added with SO2 and the one only with the plantarum. And what it is rather obvious is to see, okay, it's very complicated to get this to work. So let me see if I can do like that. I mean, do you see it like that? If I do it like that? No, it's not, because probably, okay. So I need to be like that. But this is complicated. So if you just take a look at the two microorganisms that we considered as our negative controls, or negative or bad impacting controls, that are the acetic acid and the Hansenia, you can see immediately here the effect of the SO2, because we know that those microorganisms are actually sensitive to SO2, and so we have a decrease. But you can also see that by adding Lactobacillus plantarum, you have already a decrease in the concentrations. And it is interesting to see that this aspect of the addition of lactic lactobacillus actually did not impact pretty much the other population. So if we have, for instance, non-Saccharomyces that we want to promote, because this is, we know very well that this is a trend, this lactobacillus is actually not impacting negatively on the good non-Saccharomyces, but is only impacting on the bad microbes that can affect the quality of the wine. And so you can see here, just as a highlight, that we have a stable population of lactobacillus, and then we have also a stable population of Saccharomyces cerevisiae, and at the end, the two non-Saccharomyces were actually not affected. And when we go at looking specifically at what is happening at the two populations of the Acetobacter aceti and Hansenia ovarum, we can immediately see that there is a significant difference between the control and the thesis where we added the SO2, but also lactobacillus. And so by the end of let's say, the first week, although we know that the first week is actually already too late, probably for some of the populations, but we can see that there is an immediate impact, on the negative populations. And so you can see, for instance, the orange bar that is always lower than the red bar that is representing our control. It is interesting also to see that, again, the SO2 treatment was the most effective. And so let's not forget about it, because, I mean, today we had nice interactions and nice discussions that, we don't have, by default, to move away from very established practices that guarantees quality of our wines. Yes, we need to change the way the wines are produced, but again, let's not forget about very, let's say, very established practices. And I just want to show you this slide where we can actually compare, by a simple PCA, how the different wines were placed. And it is interesting to see that, they are completely different. There is a very big difference in the two compounds we took as a biomarkers, that is the acetic acid and also the ethyl acetate. And in particular, the ethyl acetate, you can see that it was very much reduced, but also the acetic acid. And even more importantly, the plantarum was able to add new features to the wine, like, for instance, high levels of linalool, glycerol, 1-propanol, and you can read the rest yourself. So this was a very promising result that we got in 2019, and in 2020, Christian Hansen actually was interested enough to try to redo the experiment in the same, let's say, setup conditions, but adding one new variable, that was the addition of SO2. So in 2020, what we have done was not only use Lactobacillus plantarum by itself, but we also added small quantities of hydrogen, of sulfur dioxide. And again, we confirmed the trends observed in the first year, and so this is actually very important, because in two different vintages we have the same result. And we can also see that we have an impact on the populations that are not represented by the, the positive or the desired non-Saccharomyces by the addition of SO2. So when we have, for instance, L. plantarum with 3 milligrams per liter of SO2, you can see that the impact is mainly done by the SO2 because we can see a decrease. However, we still have a kind of collaboration between the L. plantarum and also the addition of SO2. And this is particularly interesting, for instance, in the 1 milligram per liter, where the populations of Starmerella and Mechnicovia were not really affected. And as showed before, if we go a little bit on the negative populations, we have a clear decrease, especially, or a clear impact, especially on the Acetobacter aceti. So this lactic acid bacteria is very active towards the control of the Acetobacter aceti because almost immediately you have a huge decrease. And remember that here we are somehow stressing the conditions because we have a huge amount of acetic acid bacteria in the mast. Okay? And the same thing somehow is also happening for the Hansenia sporovarum. So immediately we have an impact. And again, the impact here, the one that, if you see those histograms, for sure you see the effect of SO2. I mean, this is undoubted. We don't have to forget about it. But if we have also lactic acid bacteria, then the cooperation can become obvious. And last, if, again, we try to position them on the screen, the different theses, you can see that here there is more a separation that really depends on the quantity of SO2 that has been used, because you can see that practically all those theses here are the ones that have more SO2, but you have also practically the ones with Lactobacillus plantarum without SO2 and with gram per liter that are actually close also to each other. So again, we have wines that are well defined by the organoleptic point of view. And just to conclude, and then I'm just handing over to Duncan to continue on that. I mean, those are first evidences that can really underline the possibility of using Lactobacillus plantarum as a putative alternative to SO2 addition, because we can see that, especially when we are reducing a little bit the quantity of SO2, LB plantarum can really help on the control of those populations that we have been using. And there is also an important aspect on the chemical point of view. So here we don't only talk about the effect that the plantarum has on the control of some of the populations, but we also talk about the contribution that this strain had by the final quality of the wines. Of course, those are all results that have to be confirmed in small-scale production, in industrial production, but at least, we have these first evidences that really makes us believing that this can be like a feasible solution. Thank you.